think about the seedless vascular plants. So they're vascular. They have xylem and phloem, the more primitive kind of xylem and phloem. And so they have the tracheids for xylem and the sieve cells for the phloem. Now, when you are trying to classify plants and group them into, the phylogenetic tree, one of the things that they do look at is the arrangement of the vascular tissue. Like, where is it? How does it look? And what they find is that when you examine most uh, seedless vascular plants, they'll have what we call a protostele arrangement, which is the vascular tissue is like dead center of the stem and it's just all clumped together, solid cylinder right in the middle of the stem. And then the saphenostele actually, well, that one will have a pith region, which is usually parenchyma cells, and then they'll have vascular tissue around that. Usually, a saphenostele is usually pretty much a solid ring of vascular tissue. And then there's another version called the eustele-like saphenostele. So it's a ring, but it's broken up a little bit. So it's not a solid ring. It's kind of clusters of vascular tissue. But overall, the pattern looks like a ring around the pith region. So they actually look at this and they look at the arrangement, and that helps them classify the different plants. So if they discover a new plant, that's one of the things that they do, and that's characteristic. And I think you've seen this picture already, where we were looking at when we covered stem tissue just in general. And so we've already looked at monocot stem versus dicot stem. And so, but now we need to look at the other parts of this image here to think about the seedless vascular plants. So the protostele they're trying to show on the top left, which is just, a nice solid cylinder of xylem and phloem smack in the middle. And then the saphenostele, you see that ring of tissue with a central pith region of parenchyma cells. And then the eustele-like, it's usually vascular tissue kind of clumped around in a ring. Okay, so that yes, they look at that. So that's one of the things that you'll look at in lab under the microscope for these different seedless vascular plants. And then they look at leaves. They look to see what do the leaves look like, and that can help them classify too, which plants belong into which part of the phylogenetic tree. Now, when we say leaves, if I say the word leaves, you probably are imagining like, an oak leaf or a maple leaf or something like that, which is what we would call a megaphyll. Those are what you normally think of. Those are the normal leaves that most people think about. And there's been a long debate of how did these leaves evolve? Like, what parts of the plant body is it derived from? And so the current hypothesis is that you have branch tissue that started to go in a design where they're all in the same plane of space. And then they developed like webbing, parenchyma cells in between it, like webbing of cells in between to fill it in. And so basically, if you think about, I'll show you a picture of that, but if you think about my fingers on my hand being different branches, and what they're saying is that over time, the branches all went into one plane, and then it filled in with parenchyma tissue. And then that became what we see as the megaphyll or normal leaves that you think of. And I'll show you some pictures of that. The other option is a microphyll, which are technically they're leaves. They're just really simple. They're just, they think that they evolved from, and I'll show you what these enations are in a second, but enations are just these little flaps of tissue. So they think the microphylls kind of took that enation idea and ran with it a little bit more so that you end up with this small little tiny with just one little vein going into that leaf. So there's kind of two thoughts here to how leaves evolve. Because we're going to find that some leaves are technically, they're the microphyll type leaf when we look at the seedless vascular plants. Actually, most of them are. And then it's really only the fern plants, the ferns, that have the megaphyll within the seedless vascular plant. So this, it's kind of hard to describe in a PowerPoint slide, but they're trying to show this idea where you have those branches that are in different planes of space, and then they can go into one plane of space, and then they start to fill in. So that's what they're trying to show there. And then they're trying to show it here again, how it flattened out into one plane of space and then filled in. So that's how they think the normal megaphyll leaves evolved. And then enations are just these little flaps of tissue. They're not really considered leaves, but they think that enations might have been the starting point for microphylls to develop, that type of leaf. And so we're going to look at this plant in more detail too in a minute. Well, actually right now. So this is a interesting plant that is called Psilotum nudum, and it is very, very simple. You don't see, like I said, no true leaves here. There's just those little flaps of tissue. So it's mostly the stem that's doing all the photosynthesis, and then all that kind of yellow blobs that you see, that's where the spores are located, sporangium. So the common name is whisk fern, and so I do want you to keep memorizing the phylum names, please. So I'll be asking what phylum whisk ferns belong to, okay? And then you'll have to provide the Psilotum, or you'll pick from the different phylum. So the P is silent, phylum Psilotophyta, and there's only 2 living genera right now. Everything else has gone extinct. So Psilotum and Mesipterus, the T is silent on that one too. Mesipterus and Psilotum. Don't see them around here. We have one plant in the greenhouse that I think is still living. They're difficult to buy as well. They're super expensive for some reason right now. There just must not be a lot of them available. So they don't have true roots. They don't have true leaves. They just have those little flaps of tissue that we call enations. And so the stems are really the workhorse. They do all the photosynthesis, really. And so when you see that whisk fern, that's, most of what you're looking at is the sporophyte. And there's underground rhizomes, so that's stem tissue underground. Okay, rhizomes are stems that are underground. And they do form relationship with mycorrhizal fungi, the endomycorrhizal or the VAM type fungi. And then those yellow structures in that previous image, those are the sporangia. They're always clustered in 3. That's the pattern for whisk ferns. You look for a cluster of 3 sporangia, and they typically show up yellowish when they're mature. Okay, this is a term that I'm going to ask you to start knowing now, this idea of homosporous spores. So far, all the spores we've talked about, they're homosporous. But soon, there's another group of plants. I'm going to say something different. They're not homosporous. They're actually heterosporous. Homosporous means they look identical. If you look at them under the microscope, you can't tell them apart. And that's what we've been seeing so far. We saw that in the bryophytes. So when the spores germinate, they'll give rise to a bisexual gametophyte, which means you'll have both male and female gametophyte going on here. Okay, so no true roots, but they do have underground stems. No leaves, just those enations. But when you look at the gametophyte, once that starts to form, in the animal world, we tend to say bisexual, but in the botanical world, we tend to use this term, which is a mouthful, monoecious. So that's a term I want you to start knowing, because that's going to show up now. Moving forward, we're going to keep talking about this idea. So monoecious. Mono means one. Ecious means house. One house. So what they're saying there is it's one gametophyte. It has both male and female structures. This one you can't see unless you dig it up. And it associates with mycorrhizal fungi as well. It does have motile sperm, and you need free water. for the sperm and egg to unite and fertilize, and then you get the zygote developing. And then once the zygote's there, it's going to start to develop into the sporophyte. It's going to grow and grow and differentiate into the sporophyte. And we see fairly quickly that they'll separate from each other. The sporophyte will separate. And so technically they say, yeah, you know, the sporophyte doesn't really depend on the gametophyte for very long at all. They're pretty independent of each other. So the whisk fern are not a plant that we see around here. If you're out hiking around, you would not see it. It's tropical. And so here's the life cycle put into perspective. The sporophyte phase dominates and looking at the plant as opposed to sporophyte tissue. Well, all sporophyte tissue, I should say. And then you have the spores getting released out of the sporangium. And then hopefully they're going to germinate and grow. And then the gametophyte, you tend not to see it all. But they're trying to show that it's bisexual. You have both the male and female. And then once you have the sperm and egg fertilized, then you're back into the sporophyte phase. So remember, the sporophyte is the diploid or full complement chromosomes, the 2N. The gametophyte is 1N. This is from a different book, and they kind of flipped it. It's a little bit more colorful design here, but I wish they would have put the sporophyte on top, but they didn't. So whichever one you want to look at when you're studying, they're both showing the same idea. But they're really cute if you stumble upon them. If you're further south, you may have noticed them if you have been in those areas of the United States. And if you look at their tissue under the microscope, the top left image is a young stem tissue is being shown on the left there. And you can see that it's just a protostele. So the vascular tissue is just a cylinder right in the middle. And then this image here, that's just some of the, that's a sporangium, the clusters of sporangium in 3 there. You see 1, 2, and 3 all clustered together. And then this image down here is a more mature stem. And again, you can just see there's a solid chunk of vascular tissue smack in the middle. So the sporangium start out green, and then once the spores are pretty much good to go and they've gone through meiosis, those sporangia will all shift to the yellowish color, release the spores. And then the spores start germinating and growing into the gametophyte. So whisk ferns are really cool little plants, very primitive looking. They look a lot like their fossil extinct versions. They haven't really changed much. Where is, not that one. Where is it? Oh, here. So going back to this slide, with whisk ferns belonging to Psilotophyta, the other one is Mesipterus. So Mesipterus doesn't look like whisk ferns. They are much more fern-like, I guess. These are not true leaves again, and they tend to hang out on like tree trunks in tropical environments. So I think this picture was actually from New Zealand, where you can see it growing on a tree trunk, but very different than the whisk fern. But they belong into the same phylum, believe it or not. Their sporangia tissue, you can see here, they're the olfactory for the sporangia. They usually show up in pairs instead of in 3s. So this is what it would look like under the microscope. So those are all the spores that are starting to form. Eventually they'll go through meiosis. So Psilotum and Mesipterus into that phylum. And then if we shift into this phylum, so it depends which text or resource you're reading, they'll either call it Lycophyta or Lycopodiophyta. I prefer the shorter word, it's easier. But we see essentially these groups showing up into this phylum. Club mosses, sometimes they're called ground pines. We have those around here. It's very plentiful. Spike mosses we have around here. Quillworts too. So Lycopodium is the genus name that a lot of people might see around here. Solanginella for the spike mosses and Isoedes for the quillworts. So if you want to really see these, go tropical. That's where most of them are. And a lot of them, especially the spike mosses, are epiphytic. So that just means they're hanging out on other plants. They're not causing harm. They're just hanging out on those other plants. And even more coastal areas, very cold, the Arctic, you can find this phylum of plants, but we have them around here. They're very fun to see, especially in the middle of winter, poking through the snow because they are evergreen. So the sporophyte is, they do have a leaf, a true leaf, but it's the microphyll type leaf. And so they're small, and then they're spirally arranged on the stem. And you tend to see a couple of different designs. Sometimes the spore locations are kind of interspersed through the stem area, and sometimes they dedicate the tips as the place of forming their strobili. So remember, the strobili are just clusters of sporangium tissue, all tightly clustered together, and they look like cones. That's some people will say, "Oh, look at the cone on that little ground pine," but they're not true cones. But it's like the precursor to cones, really, in a way. So all the spores are homosporous. They all look the same. And again, when those spores land, they start to germinate, grow into meadophyte, it'll be monoecious, one house, or bisexual. And so sometimes it's totally underground, sometimes it's partially underground. It can be photosynthetic, depending on the species. It can associate with fungi, mycorrhizal fungi. And this one, gametophyte, takes a while. So to complete its life cycle and get fully functioning sperm and egg, it can take a long, long time, 6 to 15 years. So there's been a problem with people over harvesting this, especially the ground pines. Ground pines are really popular for like decorating, like wreath decorating, and just, you know, if you're putting together some kind of plant decoration, it's common that they'll grab some of those and they spray paint them and use them for decoration. And enough people have been doing that they're starting to get worried about some of the ground pines. So if you do see ground pines out there, try to refrain from harvesting them because they're cute, but we don't want them to go extinct or to become dangerous. So here they are, and you can see the strobili, pretty obvious. Tip here, so that's all sporangium tissue, just tightly clustered. It kind of looks like a pine cone. You see why they call these ground pines. They're really, really cute. Here you can see like the top left in the snow. If you're out hiking around, especially this year, we had not much snow at all. So the ground pines were pretty obvious when you're out hiking. Lots of different species, but they're very petite, really cute. They stay evergreen. Here's the life cycle. So we're trying to show, you just have that one monoecious gametophyte where you have the Antheridium and the Archegonium. So sperm and egg production. And then once you get the egg fertilized, you're back up into the sporophyte phase. This is from a different book, a little bit more detail, so if you want to use this for study. And they even put on here the microphyll type leaves. Okay. A couple more pictures. So here you see a lot of really healthy looking strobili. Over time, those tend to turn tan or yellowish. You have some club mosses that are more, they call this the pendulant growth form, where they kind of droop like that. And then, but again, it's usually the tip. Even if they're drooping, you look toward the tip. and that will be where the strobili are located. And then if you zoom into the strobulus tissue, that's what it looks like. And so this is the idea of homosporous spores. All the spores visually, they look the same. All right, so homosporous. And then you can see that they're nicely protected by this tissue. All right, so, and then the sporangium with all the spores inside. And then if you look at the stem tissue, interesting looking stem. So this is technically a protostele. That's pretty much just solid xylem and phloem smack in the middle of the stem tissue. Kind of a cool little design there. Okay, so ground pines, really cute plants. This is an interesting plant, resurrection plant. It's a nice name for it, Selaginella lepidophylla. This one is native to Texas, Southwest, Mexico. And if you look at it in its dormant state, it looks dead. It dries out, and then it just curls up on itself, so it looks like a ball. And probably, maybe they do that to move with the wind, kind of like tumbleweeds. So this is an interesting one because in this is a plant, even though it's a vascular plant, it has heterospores. So this is a trait seen in more advanced plants like the gymnosperms and the angiosperms. But here's an example of a seedless vascular plant playing around with this idea of being heterosporous. So I do need you to know that term too, because we're going to keep hearing this moving into the next groups. And so heterosporous, hetero means different. That means there's going to be two distinct types of spores, and you can tell them apart visually. One will be called the microspores. That's what will be giving rise to a male gametophyte. And the other type is a megaspore. Right? So the way I remember that is mega, well, egg is in the word. If it's going to give rise to the female gametophyte, which the females produce the egg. So megaspore, and they look very big. They're big, mega, big. All right. And so they produce the female gametophyte. So microspores, I always think about tiny little sperm, micro. All right. And so we're going to see these terms coming through into the next groups, because usually when we refer to anything micro when we're talking about life cycles, it usually has something to do with the male gametophyte or sperm production going on. Okay, so heterospores, spores, they look different. And so, yeah, I'll show you some better pictures here, but you can kind of see they're trying to show the microspores and the megaspores here. Okay, and then when you look at the strobulus, you will actually see obvious difference between megaspore production and microspore production. They look very, very different. Here's another image from a different book, a little bit more words on there for studying. And that one is showing the mega and microspores. So if, where is it? Interesting. All right, so here they're showing the megaspores, and then what they start talking about is the mega gametophyte. That's the female gametophyte. Right? So mega, always think about the female. The mega gametophyte female. That means like the job is to make the egg. Again, it's going to be in the archegonia tissue. Okay, each one egg for each archegonium. And then the microgametophyte, you're going to hear that term more now, microgametophyte. That's the male, and that's the job of producing sperm. So they're different gametophytes. They're separate. They're not monoecious. They're not bisexual. And so here you see a megagametophyte and the sporophyte that's starting to grow from it. So you had to fertilize. So remember now, the megagametophyte, that's where the egg is. The sperm has to get there. The egg's not moving. All right. So if you see that growth coming from that pit or sporophyte, that's the female gametophyte, because the egg doesn't shift anywhere; it stays with the female gametophyte. And then if that egg gets successfully fertilized, which it must have in this image, because now you see the sporophyte developing. And then here you're seeing a strobila, where you can see an obvious difference in the spores. And I mean, they named it correctly, megaspores. Here you see 3 megaspores in that sporangium tissue. And then you see a lot of microspores in the other sporangium being shown here, right? So heterospore, kind of unusual for the seedless vascular plants to do that. Here's again the stem tissue. So that's, xylem and phloem all smack in the middle, so that's a protostele. And then this particular species is called the shining club moss because the leaves are so shiny. It's very obvious that way. We do have some spike mosses around here. And then the quillworts are another one, the last group in this phylum to talk about. And so they're very unusual. They do, you can kind of think why they call them quillworts. They kind of look like porcupine quills, but they're not stiff. They're flexible, and they are pretty simple. Look at the base here. That's where they are forming their sporangium tissue. And so quillworts tend to see them a lot along like lakes and rivers. They really like aquatic areas. They're good food for aquatic animals, muskrats and birds, especially the base here. Very sporangium is forming. Here's another life cycle, a little bit more detail. And then if you're out hiking around and looking for them, look carefully because they can blend into the other plants in the area. So quite petite. Okay, so quillworts were the last group of that phylum, Lycophyta. Here's another one. Phylum Equisethophyta. It used to be called Sphenophyta. I certainly prefer Equisethophyta because if you remember what equa means horses, and then the common name horsetail, that fits a lot better, I think. And so we're thinking about the horsetails, which are also called scouring rushes. And they, again, they tend to prefer pretty damp, moist areas. You tend to see them along consistently moist soil. I see them in town along the lakeshore on the bike path. But you can see them along ditches, rivers, lakes, and they're really a cool looking plant. So the only one that's currently still making it, still alive, is in the Equisetum, yes. And if you look at this plant and then look to the fossil record, they look pretty much identical. They're just smaller. They used to be quite large, and now they're much, much smaller. But other than that, they haven't really changed their morphology. And so 300 million years ago, during the Carboniferous period, Equisetites was another Equisethophyta type plant, and that one was very large. So we're going to see that the sporophyte, when you're out hiking around and look at the sporophyte, they have jointed stems, and the stem tissue is very ribbed. It has a very rough texture, and it's rough because it has a lot of silica deposited in the cells that make up the stem area. And that's why they call it scouring rushes, because you can actually use them as an abrasive material. So they're kind of like a scouring pad. People use them when they're out or camping and cooking. If they burn their pots and want to clean off their pots, you can actually use the stem tissue for that. The leaves really don't look, they're technically leaves, they're microphyls technically, but they really don't look like much at all. I'll point them out in an image. Tiny, and they often look brown, so they don't really do much for the plant. And it's the stem tissue that does the bulk of photosynthesis. So they tend to have perennial rhizomes, which means every spring they'll pop up a new sporophyte. And they do have true roots. So that's something to look for in the spring weather here is perennial sporophytes will be growing up now. And they are, once you learn what they look like, they're easy to spot. So here you can see a couple of things going on here. There's the so-called fertile stem, which are these two things here, where that look a little bit more brownish, and then they have the strobili on the tip. And then the other horsetails right next to them, that's all branch tissue. Those aren't leaves, so that's branch tissue. So the Equisetophyta, they have homospore spores, so they all look identical. And they have those funky looking strobili where all the sporangium is clustered together. When you look at it under the microscope, they often say it looks sausage-like in design. And the spores actually have strips of tissue that are on their surface that break loose over time. And they think that helps again with those pieces of tissue will kind of twist during different humidity values, and that helps them release and disperse their spores. Gametophytes can be tiny, 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 or all the way up to 3 centimeters. And they're either monoecious, where they have both Antheridia and Archegonia on one, or it can just be dedicated to Antheridia only, the male only. And they have a strategy where if it's a bisexual or monoecious gametophyte where male and female are there, they stagger when things mature. And so what happens is typically they see the egg matures first, but the sperm aren't ready to go yet on that particular gametophyte. And the reason they're doing that is they're hoping a nearby gametophyte will deliver some sperm. So it's just a way to try to maximize their chance they're going to get sperm from a different gametophyte, with the idea of better mixing of genes, novel genes, and will help them survive better. So here's their life cycle from your book. So you have to look at the strobulous tissue where all the sporangium are located. So this is why they say sausage-like. Have all the design there. So that's the sporangium, and they're trying to show that's not flagella. Those are elater tissue. Okay, attached to the spores. And then the spores land and meadophytes start to grow. And you have to think about the archegonium with the egg and antheridium with all the sperm. Okay. This is another image from a different book. A little bit easier to see and maybe study off of. And a close-up of the strobulus. So kind of an ornate design for a strobulus, a little fancier than what we've seen on the other groups of plants. But when you look at the stem, these are the so-called fertile stems that have strobulus. So here, this right underneath the strobulus, you see kind of black and tan. But those are the microphyls. Those are the leaves. They're all whorled, attached at the same spot there. And then you can see another grouping a little further down here. So the leaves aren't much to think about, and it's all basically the stem tissue that's doing the photosynthesis. And then if you zoom in under the microscope, look at some strobulus. So they took a cross section through a strobulus here. So you can see the sausage-like sporangium, spores. And then when they're all good to go, they will have that elater tissue that's formed first tightly around the spore, and then as the spore matures, it starts to dry out a little bit. They'll break free, and you can clearly see the strips of tissue. If you look at those spores under the microscope, you'll see something like that. So those striations that you see, those are the elaters. So when you're out hiking around, look for them. They're really distinct looking plant, very ancient design. They haven't changed much for millions and millions of years. Sometimes you come across a big patch. This is a mixed coniferous or hardwood coniferous forest. And you can see that all of this pretty much in the foreground. That's all horsetail right there on the forest floor. And then because they live in damp areas, they tend to have not a huge amount of xylem. And they tend to have hollow central area. And then all of this is just hollow open spaces. And then the xylem and phloem are in this region here. So this is a saphanostele that you're looking at, and it's a eustele like saphanostele because you get the xylem and phloem in patches, but overall it's a cylinder. And then you can see the ribbing here on the very perimeter of the stem. So when you run your fingers over that, you'll feel that rougher texture. And there's lots of silica embedded in there, so they can be used for scouring your dirty pans if you burn your lunch for dinner. Here's a little baby gametophyte. This one's a male gametophyte, although I would not ask you to identify that, but those are Antheridia with a bunch of sperm. So not much to look at. Often you don't see this with the naked eye. It'd be too small. And then here they're trying to show that you have an egg must have gotten fertilized, and you're starting to see a young sporophyte emerging. But like I said, very often in the spring, you're going to see the sporophyte just shooting up from the rhizome. It's just going to come up every spring. Okay, so the last group of seedless vascular plants that we're going to talk about are this group, the phylum Polypodiophyta. It's also been called Pterophyta or Pteridophyta. I think your book is using Polypodiophyta, so we'll stick with that. But those are the ferns. And they are definitely the largest group of plants out there besides the angiosperms. So they even win over the gymnosperms. There's more ferns out there than gymnosperm species. So they're highly successful. But I'm going to stop there. We'll continue on with the ferns. What is today? On Friday, right? All right, we'll continue on with the ferns on Friday.